The CHIC project was aimed to see how genome editing could be of benefit to consumers in a small crop. And for that we choose chicory, which is a relatively small crop, and with small I mean grown on a little areas.
For Workbench One, our main goal was actually to find the best way to use genome editing to make mutants in chicory. So the whole project is based on making mutations and testing the effect of these, for instance, on inulin or terpenes. But of course you have to have a good method to make the mutants. And this relies a lot of lab work with tissue culture and testing different ways to actually introduce these reagents into chicory and then to look at the outcome.
So what I particularly liked is that within a very short time, within 4 years, we worked on a crop which was very little research as compared to other crops, but that we made a lot of technical progress in genome editing. We created new varieties. But on top of that, we did that in collaboration with the other pillars of the project, which is communication and stakeholder engagement.
So we really have higher quality inulin from root chicory, and we have a reduction of the bitter compounds, which makes it much more easier to extract inulin from root chicory.
So with these new technologies we can boost also the European competitiveness, because a lot of the value added stays in the European countries. And also with the higher efficiency of the process, we can reduce the greenhouse gas emissions compared to the actual situation. For example, if we are comparing it to the actual production system for inulin.
And also, of course, the other part has been related to the genome editing of the chicory plant, and we have proven that it is a kind of very feasible and good technology for different types of plants, not only model plants, but kind of actual industrial crops also.
So far that we have analyzed technically the different NPBTs used for editing the chicory, and we have found different efficiencies between the different techniques that are used, but all work very well. And we have also identified the off-target rates of the different techniques, and near to the closure of the project now, we haven't identified any relevant off-target so far, but we are still continuing analyzing the samples, and will hopefully finish this in the next months. And so far, no off-targets, which is really good.
Because we have a lot of activities still ongoing in this final phase of the project. And I think it's in about 6 months' time, we will be able to summarize the main findings. But at this point in time, it's more easy for us to comment about the observations we have made so far, in particular in more general terms on social acceptability of genome-edited plants.
Concerning the regulatory assessment is that we see a changing regulatory environment around the globe, with especially in the Americans and in Asian countries are opening up for genome editing techniques of different kinds, mostly SDN1, SDN2, and Europe is still stuck in a debate. And unfortunately, we won't see any result coming from Europe by the end of the project, but we will keep monitoring it quite closely and also continue our work after the project and keep the community updated.
Interesting thing is that we are actually focusing on two moving targets at the same time. The one is social acceptability of genome-edited plants in general. And second, within the CHIC project, of course, we try to have a more specialized focus on root chicory, whether we find factors that foster or hinder the social acceptability of genome-edited or gene-edited root chicory in particular.
The main results are that new ways of communication in scientific projects. We are used to scientific publications, we are used to conferences, posters, and so on. And the activities that we have implemented in CHIC, developing games, trying after students to make them to understand what is CHIC and so on, to work with other different type of audience and complete audience that don't know anything about genome editing, biotechnology and so on, has been quite successful and from my point of view it's a really nice result of the CHIC project.
So our goal was actually to test 4 different methods based on, what type of cell was used and how the reagents were turned on inside those cells. And so we've tested these 4 methods, and now we've come to what we think is the best method in the future to make more mutants of chicory.
So the outcomes which are related to human health are, we identified some molecules, some compounds in chicory that could have a health benefit, which could treat or prevent diseases. How to implement it and to bring that forward to the consumer, that will take another effort. But it looks very promising.